Hello my dear future doctors. Welcome back to Quick Neat Botany. In this video, let us discuss some NCERT line-based questions from Molecular Basis of Inheritance chapter. Let us start with the question. What was not clear about inheritance at the time of Mendel? Let's see NCERT lines for this question. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. The next, what conclusion was reached after nearly 100 years of research about genetic material? Over the next 100 years, the nature of putative genetic material was investigated, culminating in the realization that DNA deoxyribonucleic acid is the genetic material. What are nucleic acids polymers of? Nucleic acids are polymers of nucleotides. Name the two types of nucleic acids found in living systems. Deoxyribonucleic acid, the DNA, and ribonucleic acid, RNA, are the two types of nucleic acids found in living systems. In most organisms, which molecule acts as genetic material? DNA acts as genetic material in most of the organisms. In which organisms does RNA acts as genetic material? RNA, it acts as genetic material in some viruses. Let's discuss the viruses with RNA as the genetic material. HIV virus and TMV virus. Mention the two additional roles of RNA apart from acting as genetic material. Though RNA acts as genetic material, it mostly functions as messenger and it has additional roles as well. It functions as an adapter, structural, and in some cases it acts as a catalytic molecule. The most important function of RNA is RNA mostly functions as messenger. Next, the DNA. DNA is a long polymer of which units? DNA is a long polymer of deoxyribonucleotides. How is the length of DNA usually defined? The length of DNA is usually defined as number of nucleotides or pairs of nucleotides. What characteristic of DNA length is specific to an organism? The number of nucleotides, it is characteristic to every organism. So the characteristic of an organism is the number of nucleotides in it. How many nucleotides are present in bacteriophage 5174? A bacteriophage known as 5174, it has 5386 nucleotides. What is the number of base pairs in bacteriophage lambda? Bacteriophage lambda, it has 48,502 base pairs of nucleotides. What is the genome size of E. coli? E. coli, its genome size, it means it has 4.6 into 10 power 6 base pairs of nucleotides. What is the haploid DNA content in humans? Haploid content of human DNA is 3.3 into 10 power 9 base pairs. Next, structure of a polynucleotide chain. What are the three components of a nucleotide? A nucleotide, it has three components, a nitrogen base, a pentose sugar, and a phosphate group. Which sugar is present in RNA? In RNA, ribose sugar is present. Which sugar is present in DNA? In DNA, deoxyribose sugar is present. Name the two types of nitrogen bases. The two types of nitrogen bases are purines and pyrimidines. Which bases are purines? Adenine and guanine are purines. Which bases are pyrimidines? Cytosine, uracil and thymine are pyrimidines. Which base is common to both DNA and RNA? In pyrimidines, Cytosine is common for both DNA and RNA. Cytosine is common for both DNA and RNA, which is a pyrimidine. And thymine is exclusively present in DNA and uracil is exclusively present in RNA. Adenine and guanine are common purines for both DNA and RNA. Which base is present only in DNA? That is thymine. Thymine is exclusively present in DNA. Which base replaces the thymine in RNA? Uracil. It is present in RNA in the place of thymine. Let us see formation of nucleotide and nucleoside. Between which atoms is the N-glycosidic linkage is formed? A nitrogen base is linked to OH of first carbon of pentose sugar through N-glycosidic linkage. What is formed when a nitrogen base attaches to a sugar? Then it forms a nucleoside. What is formed when a phosphate group attaches to a nucleoside? When a phosphate group is linked to OH of 5 carbon of a sugar, then it forms nucleotide through phosphodiester linkage. What type of linkage joins the nucleotides in a polynucleotide chain? The corresponding nucleotides are joined together by 3' to 5' phosphodiester linkage. What is a dinucleotide? If the two nucleotides are joined together, then it is called dinucleotide. What gives the directionality to a polynucleotide chain? 3' to 5' phosphodiester linkage, it provides directionality to a polynucleotide chain. At 3' end, it has free OH group. At 5' end, it has free phosphate moiety. Then it gives directionality to a polynucleotide chain. What is present at 5' end of a polynucleotide chain? Phosphate moiety is present at 5' end of the sugar in that chain. What is present at 3' end of the polynucleotide chain? At the 3' end, at the free OH group of the third carbon, it is present a phosphate moiety. A phosphate moiety is present at the 3' end of the polynucleotide chain. What forms the backbone of a polynucleotide chain? The backbone of a polynucleotide chain is formed due to sugar and phosphate. Where are nitrogenous bases are located in the chain? The nitrogen base is linked to the sugar, particularly it is projecting from the backbone towards the inner side at first carbon. DNA versus RNA differences. What additional group is present in ribosugar of DNA? In RNA, every nucleotide residue, it has an additional OH group present at second position of the ribose. What is the chemical name of thymine mentioned in the textbook? The chemical name of the thymine is 5-methyl-uracil. Discovery of DNA structure. Who first identified DNA? DNA as an acidic substance present in the nucleus, it was first identified by Friedrich Mischer. In which year was DNA first identified? In the year 1869. What name was given to DNA by Friedrich Mischer? He named it as nuclein. Why did the structure of DNA remain unclear for a long time? Due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. In which year was the double helix model proposed? In the year 1953. Who proposed the double helix model of DNA? James Watson and Francis Crick, they proposed double helix model of DNA. On whose X-ray diffraction data was the model based? Based on X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, Watson and Crick proposed double helix model of DNA structure. Chargaff's rule. What observations did Erwin Chargaff make about the base ratio? Erwin Chargaff, on his observations, he observed that for a double-stranded DNA, the ratio between adenine and thymine, guanine and cytosine, these are constant and equal to 1. What is the ratio of adenine to thymine? Adenine is equal to thymine. What is the ratio of guanine to cytosine? Guanine is equal to cytosine. What does complementarity of bases allow scientists to predict? The complementarity of base pairing, it was predicted that one strand of DNA, the base pairing confers a very unique property of the polynucleotide chain. They are said to be complementary to each other, and therefore if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. How does DNA replicate according to the text? DNA replication is semi-conservative. If each strand from a DNA, it acts as a template for the synthesis of new strand, the two double-stranded DNA molecules, that daughter DNA molecules that are produced would be identical to the parental DNA molecule. Why would daughter DNA molecules be identical to the parental DNA? Because parental DNA is acting as the template for the synthesis of two daughter DNA molecules.